Welcome everybody. My name is Laida Akinfatani and welcome to my series of presentations on my case study that I've given to myself called Pharm001B. And the programs, I'm going to call them LAT101 and LAT202. So this is a second recording, but I'm redoing the first recording that I did because I noticed something that I want to share with everybody because I think it's quite important. But this case study, it's an overview of the life cycle of CAR T cells therapies activities, and it's going to be from product development, preclinical studies through to clinical trials, marketing authorization and post-approval. And it's a project that I'm giving to myself to do because I'm not in the industry for various reasons. But these programs that I've got, they're not actual real, they're theoretical, and they are not currently in clinical development or approved for marketing authorization or anything like that. But they are based on contemporary scientific advances, and they are designed to illustrate how CAR T cell therapies might be developed to treat cancers, including hematological malignancies and solid tumours, which is what I'm going to focus on. So I'm going to use these theoretical products as part of two programs. And this is my project, this is my case study, and this is something that I've devised for myself because I find it I like to be, to use my brains and to learn and to develop and to use whatever tools are out there to learn those and to progress those things. So, this is my quote, one of my starting quotes, and it's from James Joyce, and it's mistakes are portals of discovery. That when you make a mistake, don't look at it and say that it means therefore that you must give up on the whole thing, walk away, and then just say that you mustn't do anything or you have to go back to somebody who will put a chain around your neck and you will never learn at the rate that you would have learned at. It means that you have to look and see what can you discover as a result of this mistake. What is it that you have to learn? Keep going when you fall down. Get up and keep going. Don't look at a mistake as something that you have to be ashamed of, but look at it as a learning event, as something that you have to learn from. And there is something important to learn from your mistakes. And I think that this thing that I'm going to share is an example of that. It's an example of not being afraid of mistakes, owning them and learning from those mistakes and making sure that you don't make them again. Right, I'm just going to give you an overview of this project, which is one of the tasks for this particular presentation. So what I've done is I've applied the Pareto principle, where 80% of all the knowledge is concentrated in 20% effort for the work. Yeah. And that's why I try to make this whole project as complex as possible so that I can get as much knowledge in as possible. But unfortunately, I got carried away with myself. But anyway, the objective of this case study is to broaden my understanding of CAR T cell therapies through a progression of two theoretical development programs. And I aim to explore all of the life cycle activities through the following. I'm going to take two cell-based medicinal products from development through to marketing authorization and post-approval activities. Yeah, I'm going to look at all of the program activities, so the development activities, the non-clinical studies, the clinical studies, clinical trials and IND applications, and then marketing authorization applications and biological license applications, and then all the post-approval activities that occur after that. I'm also going to start looking at the risk-based approach and prior knowledge and platform-based approaches, looking at that for efficiencies, which is something that actually industry is already doing. Now that I've given this a little bit of thought, that actually industry is already doing that and taking that approach, that prior knowledge and platform-based approach in post-approval activities. It's just that perhaps that terminology might not have been used, whereas it's more used in that those terminologies are used in pre-approval, so for example clinical trials, etc. But actually the platform-based approach is what we use for a lot of activities for post-approval work. So it's not something that's particularly new or exceptional. It's just the terminology and also having certain programs that are going to be available for companies to use as a way of getting platform technologies recognized. And then I'm going to talk about the engagement with the relevant bodies. So this will be in this presentation, it will be a touching base on these. And in other further presentations, I will be looking at it in more detail. But in this presentation, I'm going to touch on regulatory authorities. I'm going to touch on health technology assessment bodies. I'm going to touch on research ethics committees and patient groups. Yeah, but I'm not going to delve into them in detail in this presentation, but it will be something that is going to be looked at across the whole of this studies. And then I'll be using a fictitious company that I've called LAF Therapeutics Limited, just to explore the options available to developers. So this is my project that I've given myself, which I am really glad to be doing, as well as some other activities. And it's something that I can and will do. And I appreciate any feedback from other people, but I'm not going to stop doing it. Right, so before we delve into the whole of the case study content for this presentation, I just wanted to give an overview of the journey that I'll be taking across the series for this case study. So this flow chart, it outlines all of the 6 milestones that I'll walk you through. From the project overview and strategic foundations right through to value demonstration, after marketing authorization and post-approval strategies, and all of the things that we have to do in between. So the first milestone will be the project overview, which I've kind of talked about, but I'll give you some more information on that. The second milestone will be putting together what I think is necessary, and that's a roadmap document, a stakeholder, regulatory and stakeholder a roadmap document. I'll be looking at manufacturing, CMC, quality strategy. So I'll be looking at the quality aspects and how you tie those aspects with the clinical and efficacy and safety of your product, how the whole thing is linked. I'll be looking at, for milestone 4, your non-clinical evidence generation, your preclinical work, your ethics, and the patient groups conversations that need to be occurring at that stage before we move on to milestone 5, which is clinical development and trial strategy. And then milestone 6 will be your marketing authorization, value demonstration, and your post-approval strategies. Now, even though I've divided these up into separate milestones, they are all interlinked. The whole objective is to start with the end in mind, to start looking at your project, but to think about where you expect to be at the end of this journey. Where do we expect to be? What countries are we planning to go for? What are there likely to be their requirements, and to incorporate those at the start of the project. And it's particularly important and necessary for these particular types of modalities. Let's have a look at my notes and see if there's anything else I have to talk about. So I guess as in my notes I've got today, we're focused on milestone 1, and this is setting the foundation for 2 theoretical CAR-T programs, and I've called them LAT101 and LAT202. And I think move on to the next slide. Right, so these next 2 slides is just going to outline the project, and this might be a change in terms of what I'm going to go for. So I might decide I'm going to go for make some changes to this slide as I evolve and as I learn more and as I develop. So the first thing is the project overview and strategic foundations. So I'll be doing my project introduction. I'll also be talking about my target product profile. That consists of milestone 1. I'll briefly be talking about platform technologies, prior knowledge and platform technologies. But I will be giving an example when I actually do the actual thing. Project management, I might talk about things, for example, what different departments might be doing and the timelines and what they have to be thinking about when. And then this risk-based approach, that's something that is applicable to these types of products, but specifically within the European Union, we have this risk-based approach for these types of modalities where we have advanced therapies because of the unique characteristics of these types of products. Milestone 2 will be this roadmap document where I've got regulatory and stakeholder roadmap, but we just need to think about the global regulatory frameworks. We need to think ahead and be aware and incorporate all of the changing knowledge, the changing requirements that each country or each region may have. So we need to have a mechanism in place to monitor that and make sure that we incorporate updates into our project and into the programs. And this is because, you know, things change, more information becomes available, regulators change and release guidance documents or legal frameworks and regulations. So we do need to make sure that we're up to speed. And then we've got the stakeholders engagement, and we need to think about all of the stakeholders that are involved. So it's not just the regulatory people, it's the patient advocacy groups, it's the health technology assessments bodies. You know, we've got to make sure that we think about them not as we have to deal with them, but prior to. Be prepared and think of the end at the start of your project. Milestone 3 relates to the manufacture. It really relates to the quality aspects. So we're talking about process development, looking at the two different models, the autologous versus the allogeneic. We're also looking at the CMC and analytical strategy, raw material sourcing, analytical characterization, comparability protocols, and process validation. And then we're looking at GMP compliance. You know, we have to comply with good manufacturing practice. We also have to comply with good tissue practice as well. So we need to consider all of these regulations and all of these requirements, legislation, and then the advanced manufacturing considerations. So automation is something, as much as possible, we want to implement. Automation means that we have, you know, we build into our processes consistency. We reduce the likelihood of contamination. We make our product utilize the most up-to-date, cutting-edge technology. We want to think about things like digital manufacturing, and we also want to think about future expansion planning. And then, as I said, global regulate CMC requirements. You have to address the different CMC requirements and harmonization challenges, and I think that should be addressed at that stage. This is my second slide, which details the last 3, and I'll be looking at milestone 4, which will be the non-clinical ethics and preclinical evidence generation. So here we'll be looking at the non-clinical requirements and the guidelines. So the objectives of your non-clinical studies is to demonstrate proof of principle, or proof of concept. You need to define your pharmacological and toxicological effects that are predictive of the human response. So you want animal models that are predictive of what will happen in the human being. And so and this doesn't just occur prior to your initiation of your clinical trials, although the most crucial and critical work has to be done prior to your clinical trials, because those are what you're going to be using to get your clinical authorization. So you do need to do your preclinical and non-clinical studies, and you also need to ensure that you get buy-in from the regulators with regards to your non-clinical strategy to make sure that it does inform on your clinical goals. But basically, your non-clinical studies, your goals are to provide information. You want to provide information on what the safe doses are for your clinical trials. You want to provide information on your route of administration and your scheduling. You want to provide information on, for example, exposure and potential follow-up timelines, with regard, and that's particularly important for these kinds of products. And you also want to be able to detect or understand adverse reactions. So these things are really critical before your clinical trial begins, and you want to get those in line and understand those and be able to get the buy-in from the regulators with regards to the fact that what you're going to do, what you're going to spend time and money doing, is going to actually support your clinical trials. And then your ethical review and informed consent. So this is really important to start looking at this at this time frame. Your ethical committee, the ethics committee, you don't want them just sort of finding out about it when you're making your application. You want that early engagement. It's your ethical review is fundamental, and it's fundamentally tied to your clinical trials. You can't get one without the other. So it's there to protect human research participants. So it's important to include it at this milestone, I believe, because it's an essential interaction, and you need to get that alignment and agreement with the ethics bodies in order to streamline your clinical studies and your clinical trial applications. So it's important to be making those studies at that point. And then what else have I got? That's it, really. I think that's it for milestone 4. Milestone 5 is your clinical trial authorization. So there are various pathways in the UK and in the EU. In the EU, it's the clinical trial regulation. In the US, it's your IND applications. And then there'll be other global documentation and global requirements. So how do we design our clinical trials? How are we going to, what's the approaches that we're going to use? What about our pediatric input, hospital exemptions? What expedited pathways can we use? All of these things are going to be part of our plan, part of our considerations, and we need to incorporate them, I believe, at that time point, which is your milestone 5. And then your milestone 6, which is your marketing authorization. So by this point in your development program, you would have all the information necessary. You would have followed a strategy which involves continuous collaborative engagement with the relevant health authorities and the relevant bodies in order to ensure that you get your product authorized and that you get your product such that it's available to patients who need them, where there is an unmet need and where the quality of life will be significantly improved, or the life expectancy would be lengthened. So you want to ensure that you have all your ducks in a row. You want to ensure that you have all of the requirements by the time you've got to your marketing authorization, so that you can progress it in a streamlined fashion. And then your post-approval commitments, because you probably will have some due to the nature and due to the patient population for these particular medications. So you need to probably have some kind of management system and some kind of regulatory or some auditing way to ensure that you meet your commitments. And pharmacovigilance is something that is applicable to all it's applicable to all medicinal products and all modalities, but specifically for these types of products, it's going to be a little bit much more involved and intensified because of the fact that the persistence of these drugs are likely to be lifelong. These changes are lifelong changes and therefore require a longer monitoring period. So I just want to make a start, and I want to talk about the approaches that I made in my first presentation, which I've now taken off, and I want to address the challenges and the reason why I went for what I went for, the very, very ambitious approach that I wanted to go. So what I wanted to do was to share this journey where I'm taking the Pareto principle and I'm using it to learn as much as possible about a subject matter. And I got very ambitious. I had my 2 development programs. I had LAT101 and I had LAT202. LAT101 is the ORGate, and I was going to have this dual binding thing, and the LAT101 was going to be via the lentiviral route. So the lentiviral vectors will transduce the patient's T cells and you would get this kind of a thing. Yeah, so I think it's a good idea, and it was great, and I was really enjoying it and getting a bit carried away and a bit over enthusiastic, where I had this dual antigen binding and dual co-stimulatory domain. I thought that's great, and I thought I can get as much in as possible, and I can cover as much, and we can learn as much. However, in reality, this is just not possible. Yeah, it's not possible because your lentiviral vectors are not going to be able to have the payload. They're not going to have the space, the capacity to take in your CD3 zeta, your CD28 41BB, and the whole of the architecture of each of these CAR T cells are not going to be able to be delivered in a lentiviral vector. It won't have the payload. But it was a nice idea. It was a nice idea, but I think I was just getting carried away with myself. And it's the same for the second vector as well, that you're not going to get that the payload, the vector payload, isn't going to be able to account for this kind of genetic expression cassette. So this was my first idea, but it's not really doable. And in addition to that, your manufacturing, how are we going to ensure that we can manufacture this product where you don't get dominance of one viral vector in your T cells against the other? We can't. So I've just decided that, it's just not possible. And so I've just shelved that. But that's a good learning point, something that we learn. You make mistakes, you learn from your mistakes, and you move on. You don't say that now that means that you have to stop and that you can't move, you can't learn from it. You learn from it, you share your learning, and you move on. Then I decided, I'm not going to be able to get in 2 co-stimulatory domains. I can just have the one. And that's great. One lentiviral vector can take it. But then the more I started to think about this, the more I realized that this is just not feasible, because your starting material is going to be your patient's T cells, and there's going to be variability in that as it stands. You have to have a lot of control over the over your your manufacturing process to make sure that you don't get this over dominance with one over the other. And I just felt, and it may very well be possible, that this route is a possible route, but I'm at the start of this project and I'm thinking, of potential problems. And the more I think of it, the more I think this is going to be very difficult to be able to control. These are living cells and they are very sensitive to changes, and it may very well be possible to do it this way, but I feel that there will be problems with regards to manufacture. However, with regards to consistency in manufacture and ensuring that you have, the same proportions of the different CAR T cells. So I just think that this is not the way that I want to go. So this is going to be my final approach, which I didn't really. I knew about this approach, but it wasn't one that I had. I thought would be. I thought it would be too complex and too complicated. But I think that this is the final approach that I'm going to go for LAT101, where you have this OR gate, and this is this tandem approach. And I feel that this way we can address the issue with the vector payload, and you can address the issues with the you don't have the issues with regards to manufacturing inconsistencies with regards to the two different viral vectors. I think it's the only way where you can get two binding domains. But there are issues, I think, with this, and this is the reason why I didn't go through it to begin with. And the issue was with regards to the steric hindrance and the fact that we need the antigen binding site to actually bind with the antigen, and potentially there might be problems with this steric hindrance. So with the way that the access that the binding domains may or may not have. So excuse my very childish, if you like, drawing, but this is the approach that I'm going for, the tandem CAR T cell approach, where you have your two binding sites and you have it's all delivered in the same vector. So this is just a slide that demonstrates or just talks about what the issues were, and this is the issue of this vector payload for LAT101, and it's just not, we don't have the space to do what the original the original concept was. We just don't. The problem with taking a theoretical product and having a mistake in it is that later on that the mistake will compound and you'll get more problems as a result of it. So the strategy that I've decided to go for is this tandem approach, where we have one co-stimulatory domain as opposed to the two, and we just have the one viral vector that delivers this particular genetic recombinant nucleic acid sequence. LAT202 doesn't have the same restrictions, so because of the fact that this method is going to involve induced pluripotent stem cells, gene editing. So we can edit in the cassettes into different areas of the genome, and we're not dictated to by limitations with regards to our vector. So this is different. The only slight issue, I think, relates to where do we put these because we've got the genome, or the loci, if you like, for the T cell receptor. So this would be the endogenous T cell receptor, and we would put one of the chimeric antigen receptors in that position. So where do we put the other one? But there are other safe harbor positions that we can put the other T cell in, and it will be fine. So for LAT202, I think we can go for this format, and I think it is feasible, and that's what I'm going to go for. The considerations are the normal considerations and problems that you are likely to have with the use of induced pluripotent stem cells, and with the issues with regards to differentiation. Oh, I've got some notes here. I think I just made some notes with regards to the domain, the transmembrane domain, and just making sure that the transmembrane domain works with regards to the type of what they're binding to. So this whole structure, architecture, they have to be, the domains have to be compatible with one another. And if you're using 41BB, then CD8A is a good transmembrane domain, as regard to the hinge as well, or the spacer. CD8A is a good example. So you need to ensure that you've got the right domains that are compatible with one another, and you need to provide all of that information in terms of the nucleic acid sequence in your characterization section of your dossier. Oh, and here's just another, I think I was getting carried away, another diagram, and really the point that I wanted to make is that for this particular LAT202, it's more than just the T cell receptors, because this is dealing with solid tumours. And for solid tumours, I just realised something. For solid tumours, you have this tumour microenvironment that you have to overcome. And that's been the problem with traditional CARs, is that they've not been able to overcome these tumour microenvironment. Therefore, we've introduced other receptors as a way of boosting the efficacy of these CAR T cells. And basically, this is this TGF-beta receptor and this CXCR2 receptor. So the TGF-beta receptor is useful because TGF-beta is an immunosuppressive cytokine. So tumours often express this TGF-beta. Yeah, and basically they create this immunosuppressive environment. There are other factors as well that are responsible for creating this immunosuppressive environment, but TGF-beta is one of the major components that's responsible for that. And basically what it does is it just turns off or deactivates the T cells, and therefore the T cells become bystanders and are not able to. And basically TGF-beta, this cytokine, is part of the immune homeostasis or cytokine. It's important in ensuring that balance, that immune activation balance, where you are at the right balance, where you can address and attack pathogens or cancers, but not so much that your T cells then turn on healthy cells and you get this autoimmunity issues. So TGF-beta is crucial for maintaining that immune homeostasis and preventing autoimmunity by inhibiting, expansion and functions of various immune cells, including T cells. And the problem is that the tumor microenvironment overexpresses this TGF-beta and creates an immunosuppressive environment, and therefore the T cells are not able to attack and kill off those tumor cells. The aim is to overcome that by connecting this TGF-beta, so knocking out the TGF-beta endogenous receptors in the endogenous genome and inserting this TGF beta with an activation domain. So it's activated as opposed to inhibiting, and therefore, or co-stimulating, and therefore, I think it's co-stimulation that we'll be going for. And therefore, instead of the TGF beta being an immunosuppressive issue in the tumor microenvironment, it becomes activated. However, with a caveat for that is that there's likely to potentially, we're looking at potential side effects with that approach is what I'm thinking of off the top of my head. But we've also got this CXCR2, which is responsible for trafficking these T cells to the tumor. So hopefully that occurs where the tumours are, and hopefully avoiding the healthy tissues. So one of the things that I've noticed in industry is that sometimes departments and stakeholders are very fragmented, because you've got different specialities all working away, doing a very good job in their area of expertise. And there is a need, I think, to bring them all together and ensure that everyone is aligned. And we all need to understand and read and understand each other's roles and our responsibilities and how we interact with one another and what the communication pathways are. So this is milestone 2 that I'll be drafting this kind of a document. But we kind of need some kind of life cycle management manual. I think I decided to call it the life cycle management and roadmap manual, but I think I might change the name at some point. But it's just there to make sure that all the available stakeholders are all referring to the same document, because it's going to be something that's a living document. It's going to be something that is version controlled, and it's going to be something that ensures that we're all pulling in the same direction. All of our resources and the energies are effectively directed, and I think I've already got that there. It ensures that, even if you have a change of staff, because these products are long-term products, and you're not going to have everybody working in the same company for the rest of their lives, so you're going to have changes in staff. You need to ensure that all your stakeholders can immediately understand the vision, the status, what happened in the past, and what the long-term goals are for the products. And it provides that transparency to all concerned in the project and ensures that all the departments and the stakeholders are aligned. So this is the LAT202. I don't know why I've got it here again, but I think I've already explained. So this is the product description. I don't know, did I put everything here? Yeah, so it is a autologous or-gate tandem dual-specific CAR T-cell therapy targeting CD19 and CD22. So it has one co-stimulatory domain. It has this inducible caspase-9 safety switch, which is introduced via gene editing afterwards. It's intended for the treatment of relapsed or refractory B-cell malignancies, such as diffuse large B-cell lymphoma, DLBCL, and acute lymphoblastic leukemia, ALL. And the therapy aims to improve B-cell targeting by allowing T-cell activation upon recognition of either antigens, thereby reducing the risk of antigen escape. So prior to that, we'd gone for the CD19, but some of the B-cells then underexpress CD19. Sorry about that. And therefore they escape, and we get the malignancy again. So the cancer again comes back. So if we have 2 targets, so we reduce that risk of antigen escape. So it's going to be autologous cells, autologous D cells. So it's the donor and the recipient are the same, so that reduces the potential for graft versus host disease. Well, reduces it. So we have this dual specificity. We have CD19 and CD22, one co-stimulatory domain, remember, because of the vector payload. We have the safety switch. So this is this inducible caspase-9. and the gene editing is relates to the safety switch incorporation. Right, for LAT202, this is for solid cancers, yeah? So what have I got here? So it's allogeneic. So this is going to be, the aim is to get an off-the-shelf product. So it's going to be allogeneic. So there's going to be certain challenges involved in that approach. But there are certain advantages with having an off-the-shelf product in regards to the vein-to-vein time frame, specifically and particularly for patients that are dealing with aggressive forms of cancer where they need the treatment as soon as possible. So, and also where, for example, with regards to dosing, if you want to have a second dose, you've not got those issues with the time lags. So there are certain advantages with going for an off-the-shelf approach, but then there are going to be certain safety factors to take into consideration. But anyway, so it's going to be an AND gate, dual specific CAR T cell therapy targeting. So this is this aberrant glycosylated MUC1. So MUC1 and HER2. So MUC1 is mucin 1. So MUC1 is the aberrantly glycosylated. In normal cells, this protein that covers the cell is highly glycosylated with carbohydrates, and the surface is covered that you don't have you don't have any of the protein or the peptide residues showing. However, in certain tumor cells, this MUC1 is aberrant glycosylation. So that means that some of the proteins is showing. It also means that the carbohydrates isn't fully covering the molecules, and the carbohydrate is, you get this truncation basically. So it's what we refer to as a tumor-specific antigen. HER2 is a receptor protein, and it's expressed at low levels in some normal cells. So some epithelial cells in the breast, gastrointestinal tracts and other places. However, many cancers overexpress HER2, due to gene amplification, which is associated with aggressive tumor behavior and poor prognosis. So to focus on HER2 overexpressing tumors, is a good approach. It allows selective targeting, and if we have this AND approach where we are also this AND gate where we also have to have both the MUC1 and the HER2, if we have that approach, we introduce this much more precise precision product. This sort of precise product now means that we reduce on-target off-tumor toxicity, which is what we want to, because of the fact that HER2 is a receptor protein that's expressed at low levels in some normal cells. So that's the reason for going for this MUC1 and HER2. But MUC1 isn't what we're targeting isn't normally found in healthy tissues. This aberrant glycosylated MUC1 is a tumor-specific variant, and it's of the MUC1 protein, and it's not found in healthy tissues, or is present in different and non-pathogenic forms. So this abnormal glycosylation is what makes it cancer-specific. It creates these cancer specific epitopes, and that makes it highly tumor specific antigen. And I think I've done some literature studies, and it that confirms that both the overexpression of this aberrant tumor-associated glycosylation are hallmarks of cancer-associated MUC1, and the combination of that makes this a highly tumor specific antigen, and this is a good rationale. I thought. So what else have I got here? Yeah. Oh, I don't think I talked about the... Oh, yes, I have. Yeah. And that's the gene-edited TGF-beta mediated immunosuppression. So basically, we have flipped this this TGF-beta, so it's conferred resistance to the TGF-beta mediated immunosuppression. So what that means is that instead of getting this suppression of activation, what we're doing is we're activating, or we're stimulating the cells, and we're not getting we're using the tumor microenvironment against itself, as opposed to what it would normally do, and so therefore we're enabling activation of those T cells, and not deactivation. And then we've got these chemokine receptor CXCR2, and that's to facilitate tumor-directed trafficking. And LAT202 is intended for the treatment of advanced or metastatic solid tumors that overexpress HER2 and overexpress aberrantly glycosylated MUC1. And the AND logic gate ensures that T-cell activation upon simultaneous recognition of both tumor-associated antigens, enhancing specificity and minimizing off-tumor toxicity, which is something I've already said. So yeah, this is what the challenge is for this, and that is that it is an allogeneic T cell, so it's going to be off the shelf. You've got this dual specificity. You've got two different co-stimulatory domains, although the domains are going to be for, one for each, one for the MUC and one for the MUC1 and one for the HER2, and then you've got this TGF-beta, oh, that should be a beta there, sorry. TGF-beta has been engineered, so instead of being inhibitory, it's now activating or stimulatory. We've also introduced this inducible caspase 9 and the tumor honin. Yeah, so this is a carryover from a previous presentation. There are some similarities in the architecture. I think I shouldn't have included this. There are some similarities in the architecture. So you're binding two domains, but they're not similar in other respects. So I think this slide really shouldn't have been in here, but it is what it is now. It's here now. And as I said, the justification is to address this antigen escape, which is common and a problem for some leukemias. So you want to have the dual approach where you have the CD19 and the CD22. But remember that this is a tandem binding as opposed to two different CARs. So it's not really the same architecture. Or there are similarities, but it's not identical. And basically, it's just this redundancy-based dual targeting. So if CD19, once all the CD19 is gone, or if it gets a CD22 instead of a CD19. And LAT202, this is indicated for solid tumours. And here again, we're really concerned with precision. We want to reduce this toxicity, this off tumor toxicity. And I've already talked about the TGF-beta synthetic receptors and the CXCR2, which directs the immune cells towards tumor by following the chemokine gradient. Oh. Yeah, so this is just a part of my explorations and investigations. There is another way of doing the AND gate, and that's via this synthetic notch receptor, where you have this sequential activation you activate the first antigen, and that triggers the expression of the second one, but I haven't taken that approach. Right, so that's what the products are. And the next thing I want to talk about is prior knowledge and the approach that I had planned and will utilize to a limited degree, where we take knowledge that's already established in industry or already established publicly available, established scientific principles, and we document it in a consolidated application, and then therefore we can then rely upon that information for subsequent applications. So prior knowledge encompasses all the relevant scientific, technical and experiential information that a company or the broader community has accumulated from previous developments and manufacturing activities on similar products or similar compounds or processes. So it includes publicly available literature, established scientific principles, etc. Really, prior knowledge can really, in industry, it really involves sort of the knowledge that people have when they're manufacturing a product. Say, for example, a very simple product like a pain injection. If a company has experience with managing multiple similar products and they want to apply that experience to another product, or that knowledge to another product, without having to generate all the data, they will rely upon the experience from that they have accumulated from the other product. So it means, say, for example, that they can implement reduced testing, say reduced in-process control testing, because they see that it's not necessary and they've got a lot of experience to back it up. So that's kind of what actually happens in industry. What have I got here? The knowledge is used to inform and support developmental decisions. So yeah, if it is at the developmental stage, but it's also to reduce experimental burden, because you don't need to do all the testing, because if you can justify it, if you can say that there's no changes in what you had done before, so you have to have a risk-based approach. But it also facilitates your regulatory submissions, and it facilitates it makes everything more efficient, and it means that the regulators can review and approve something. They don't have to re-review data that they've already seen, or they can rely upon, or they have confidence on your historical knowledge or your knowledge on this particular thing, because you're experts in this area. So I've given some examples there. Platform knowledge, then that's a subset of prior knowledge. And basically the difference with platform knowledge is it's a group of technologies, or modular, and that's the most important thing. It's modular. It can cut it up into different into a component, if you like, and you can apply that component in other for other products, for other technologies, also for similar products, I guess. So it's a platform knowledge refers to especially to a subset of prior knowledge that relates to a defined group of technologies or modular elements, and it's a standardized technology that can, and that's the point. It's modular and it can be repeatably applied with minimal changes to multiple products. So it does often involve proprietary and developed in-house knowledge within the company or contract organisation, and it may include analytical procedures, manufacturing steps, safety switch technologies that are consistent across product candidates. And that's the purpose. The purpose is for efficiency. You want to be able to leverage repeatable technologies, repeatable modular technologies to accelerate your development and to establish that regulatory familiarity, which you would have done, and enable efficient scale up. And again, there's some examples of that approach. So this is a draft guidance at May 2024, and at this date that I'm looking at, it's still a draft guidance. It hasn't been implemented, but it's basically a designation program. So platform technologies in the United States, they want to have this program where you can designate a particular platform. So you'll have a platform master, a master file, just as you have active substance master files. So this guidance details the eligibility factors for receiving platform technology, talks about the benefits, how to leverage the data, how to discuss your planned requests, and what the contents of your designation request submission should be. So it's a proposal. I don't know whether it will be implemented. I may perform a theoretical application for this. I'll let you know what my results are. But this program is intended to result in efficiencies in drug development, manufacturing and review processes for drug product applications that incorporate designated platform technologies. There's an empty slide here, and the reason for this empty slide was because I wanted to put down something with regards to microRNA and the approach that the European Medicines Agency took for microRNA, but I didn't get around to it. Right, I just want to talk about platform technologies and prior knowledge because it's something that we've been dealing with for a long time. It's just that I don't know, I never really thought about it in that context. But there's nothing unusual about this. We've had active substance master files for decades, where we apply these active substance master files for chemical entities. We apply them to different products. It's the same dossier that's already been reviewed. There may be product specific requirements, in which case we'll have a bridging report, and the company that owns the active substance master file will provide the necessary documentation. But it's already embedded into our regulatory approaches and procedures. We already have active substance master files. We already have drug master files. We actually have certificates of suitabilities, and we have TSE certificates of suitabilities where the dossiers have already been reviewed by, in this case, the EDQM, and they don't require re-review. You just simply include it in your dossier application. You make your application, and you get your application hopefully approved. That those depend upon your active substance, so your drug substance, but it's not something that has just occurred or we're just looking at it. It's a concept that's already available. It's a practice that we're already applying. It's just, I don't know, I think I was just thinking in terms of advanced therapies, but actually it's something that's already done in industry. And it's just, as I say, it's an approach that improves efficiency and reduces the requirement to re-review data. And if we have active substance master file, I don't know whether it's still the case, where different regions are now accepting they're accepting the review of another safe agency. So, for example, another country may accept the European Medicines Agency's review of an active substance master file, and therefore they may not re-review it, or they may only review it where it's applicable to their own country-specific guidelines and requirements. So, and this is this concept known as regulatory reliance, where other regulatory health authorities rely upon the assessment reports from the competent authorities, or the health authorities, or the regulatory bodies that they trust and they can accept. So these are bodies like, for example, the European Medicines Agency, the FDA, and Health Canada. But remember that the reliance process is a tool. That should be a tool to inform and streamline. But the regulatory agencies themselves, they don't just simply rubber stamp the other decision. They are fully responsible and accountable for making their own decisions. It's just a tool that is used to inform and streamline their own decision process, but it's not there to outsource. They're not outsourcing, they're just including efficiencies. So this is just a diagram that I've got that shows where we can share, what we can potentially share and what we can't. Obviously, it's just showing the programs, and what is similar, and perhaps where there might be overlap, but what we're looking to do. And the aim is to start them both at the same time, but LAT202 program will take the longest, and therefore we're going with this LAT101 in order to get approval and to provide some kind of clinical credibility. And this is the point is to this slide is basically to outline the overall development strategy, and why we've chosen to start with LAT101. I say why I've chosen to start with LAT101, which is the autologous program, before moving to LAT202, the allogeneic induced pluripotent stem cell based product, with regards to the regulatory submissions. Both of the developments are going to start at the same time, but we're taking this phased approach where we're going for products that we already know are, we've got that credibility, and that we can do it quite quickly. So we get that clinical credibility. We want to be able to prove the biology in a well-characterized, regulatory-friendly disease setting. Where we can, we will use this platform approach. So something that's already been approved and reviewed, we'll just refer back to them, include in there our comparability or bridging reports. Obviously, we have to have two different applications for these two different products, because they're two different products. But as I say, it allows us to have this more time, if you like, to develop the induced pluripotent stem cell derived products while under GMP autologous program. And this is another point, and that is to build this regulatory confidence through early engagement. So this program approach that we're doing, where we are looking to utilize the information from LAT101 to LAT202. Why are we doing that? Well, is that approach acceptable to the regulators? We need to just make sure that they have the same buy-in, that they agree in this situation, in this scenario, we can utilize this information. They can rely upon the data that we've already provided them, say, for example, for analytical methods. So we just want to make sure that we get that early agreement and early engagement, and we get that engagement with regards to the products that we're designing. Do they agree with our philosophy? Do they agree with our approach? So we want to have that early engagement detailing our approach and our programs and why we're doing what we're doing, and just ensure that the data that we're going to generate is going to be suitable for what we're planning to do. And this is kind of an overview, if you like, about the interactions with the regulators. I've just put one or two. I haven't put all of them. I've mainly focused really on Europe, UK and the EU. But really what we want to do is we want to be having regular meetings. We do not want to just be going through submitting an application, which I've done in the past, where we've submitted an application, then we've come back, we've got all kinds of questions because we didn't get our strategy reviewed with the regulators first. So we want to make sure that we have that, not just that early engagement, but that continuous engagement with the regulators to ensure that, and not just the regulators, but with all of the stakeholders. So that would be the ethics committees or the independent review board. It would also include the patient advocacy groups and the health technology assessment bodies. So we want to make sure that we're not just talking at them when we're scheduled in, but we have that early engagement. We have those talks and we generate the data that's required, so that we're not going back and generating data. We've generated it at the time, and we can have that smooth, streamlined approach. And I wanted to end with a quote And the quote is that success is not final, failure is not fatal. It is the courage to continue that counts. And that's very important, that one should not be afraid of making mistakes. One should not be afraid of doing something just because the people who are standing by you are saying that you mustn't do it. You shouldn't do it. That's more of a driver to actually do it. Success is not final. Even if you succeed, it's not final. But failure is not fatal. It's the courage to continue that counts. I just wanted to give a list of my references. Here they are. Some of them, I haven't put all of them down. Right, so that's the end of my presentation. I've got a second presentation on milestone 1 because we have to go through the target product profile and we also have to go through the risk-based approach. Please don't hesitate to leave any comments in the comment section, and thank you so much for your time. I hope to hear from you again. Bye bye.